Hello everyone, welcome. This is variant 3 of May June 2022. Before we get started, make sure to check out the free notes I have available for you in the comment section. If you are new around here, this is my channel where I offer you everything from tips and advice to guided solutions to specific past papers. So if you want anything in specific, whether it's tips or, for example, a specific paper that you want to see solved, make sure to ask this in the comment section. And also, if this is possible for you, like and subscribe so that we can share these videos to as many students as possible. Let's go ahead and get started. Question number 1. The diagram shows a section through epithelium found in part of the respiratory system. Calculate the magnification. Sure, if you want to calculate the magnification, you have to remember our famous triangle. Magnification equals to image length over actual length. So this is our triangle over here, MIA. Okay, now in this case, they gave you the actual length of the cilia. Of course, no image length. We wouldn't be able to see this if it was 80 micrometers. So the actual length is already given to you. You need to calculate the image length. If you want to do that, you're going to use this scale over here. So the actual length would be 80, but the scale over here is actually 4 centimeters. So since it's 4, that means it's 40 millimeters, because 1 centimeter to millimeter you multiply by 10, and then we need to convert that to micrometers so that they're in the same units. So then from 1 millimeter to micrometer, that's 1,000 units. So now 40, you cross multiply, and you have to multiply by 1,000. So you basically are going to have 40,000 micrometers. So now we divide 40,000 by 80 using your calculator. You will get 500. Obviously, my calculator is not very accurate. I mean, my ruler is not really accurate. So I would assume that it would be 350. That's the closest to what I got. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. 1 mRNA passes through to the ribosome, 2 synthesis of polypeptides, 3 packaging of hydraulic enzymes that will remain in the cell. The appearances were listed by letter. B. Membranes which surround an enclosed inner cavity. W. Non-membrane bound spherical structures. X. A double membrane interspersed with pores. Y. Non-membrane bound cylindrical structures. And then Z being membrane bound sacs arranged as a flattened stack. Okay, so which students correctly ordered or matched the numbered functions with the appearance of the cell structure? Let's see. Okay, I would assume that mRNA passes through to the ribosome. That would be like on the rough endoplasmic reticulum, right? So the mRNA will come onto the ribosomes that are on the rough endoplasmic reticulum. So that would be membranes which surround an inner closed cavity. Okay, because that's just a description of the endoplasmic reticulum, rough endoplasmic reticulum. 2. Synthesis of polypeptides. I would assume that's the ribosomes, and these are non-membrane bound organelles. So we'll say non-membrane spherical structures, that's W. And then packing of hydraulic enzymes, I would assume this is the Golgi apparatus. So that's going to be a single membrane organelle. Okay, so it's a membrane structure which also surrounds an inner cavity. So, but let's see the options here. We have VXY. Y being non-membrane bound cylindrical structures. I would immediately cancel this. Okay, so this is wrong. And then VZZ. So we have membrane bound sacs arranged as flattened stack, but the ribosomes are not really membrane bound, so I would say no. XWZ. We have X double membrane interspersed with pores. Well, I mean, I could assume that this is like the nucleus, but you know, it could work actually. Yes, the nucleus is connected to the rough endoplasmic reticulum, and the mRNA does pass through the nuclear pores to get to the ribosomes. So that is true actually. Okay, so yeah, I would assume this is the nucleus they're talking about. Okay. And then 2 would be W being non-membrane bound spherical structures, true. And then 3 being Z, non-membrane bound sacs arranged as flattened stack. I would assume this is the Golgi apparatus, so this will be C. Okay, 3. An experiment was carried out to separate the cell structures in an animal cell. The cells were broken open, the extract was filled and put into a centrifuge tube. The tube was then spun so that the heaviest cell structure sank to the bottom, forming pellet 1, as shown. And I would assume that the first pellet would be the nucleus, because the nucleus is the heaviest cell organelle. The liquid above pellet 1 was poured into a clean centrifuge tube and spun again at a higher speed to separate the next heaviest cell structure. This cell structure sank to the bottom, forming pellet 2. I would assume that would be in the mitochondria. The procedure was repeated twice to obtain pellets 3 and 4, each containing a single cell structure. What is the function of the cell structure extracted to pellet 1? Okay, in pellet 1, so since I assume that would be the nucleus because it's the heaviest, I would say that the function would be, for example, production of mRNA, cellular division, and everything else. So we would say production of mRNA, that's C. ATP molecules are synthesized in the mitochondria. Which sugar is found in these ATP molecules? Okay, so which sugar is found in ATP molecules? It's going to obviously be a pentose sugar. Specifically, it would be ribose, not deoxyribose, so we cancel this. It will not be a hexose sugar, so we could cancel these guys, and so it will be a pentose, because ATP is specifically a pentose plus a nucleic acid base, and 3-phosphate groups. Now moving on, we have which row shows a comparison that does not correct between a typical prokaryotic cell and a typical eukaryotic plant cell. Okay, a prokaryotic cell is DNA is not associated with histone. That is true, so this is correct. And the eukaryotic plant cell, this is a correct comparison. And no endoplasmic reticulum, endoplasmic reticulum, that's also a correct comparison because prokaryotic cells do not have membrane-bound organelles. Peptidoglycan cell walls and then cellulose cell was another correct comparison. Lastly, all ribosomes are approximately 80 nanometers. All ribosomes approximately 2 nanometers. This is a wrong comparison. The reason for this is because plant cells do have both 70S and 80S ribosomes. What that means is they have both ribosomes of 80 nanometers and 22 nanometers. So I would say this is wrong because, again, there are 70S ribosomes in plant cells, such as in the chloroplast. So I would say D is my answer. Okay, it is suggested that primitive prokaryotic cells may be ancestors of certain organelles in eukaryotic cells. So which organelle is most similar in composition to a typical prokaryote? Actually, that would be the mitochondria, because just like the mitochondria, prokaryotic cells have circular DNA and they have free F70S ribosomes and only 70S ribosomes. So I would say C, obviously not the nucleoli, not the lysosomes, not the Golgi bodies. Okay, 7. Concentration of reducing sugar in a solution can be found if an observational measurement is compared to a standard. Which observational measurement could be used to estimate the concentration of a reducing sugar in an unknown solution? The color of the solution after 20 minutes, that could be possible. We could use this information. The time for the first color change to occur would also be one of the ways we do the test. The rate of formation of solid particles, it actually takes quite a while to form solid particles, so maybe not. Okay, especially it's not like a really accurate way of measuring. It's not like you can detect them, especially since the color could be dark and stuff. So I would just cancel out 3 and say 1 and 2 only B. The diagram shows 3 hexose sugars. Which row correctly shows an example of carbohydrates in which these 3 hexose sugars occur? Okay, so we have 1 being down, down, up, down. So this is alpha glucose. Okay, and then for here we have the hydroxyl group being 1 up, down, so this would be beta glucose, and this would be found in cellulose. And then we have here fructose. So I would say fructose is found in sucrose, alpha glucose is found in sucrose and the lactose, and also beta glucose is found in cellulose. So I would say 2 is definitely 2, cellulose is definitely 2, and then 1 could be true, but then 3 is wrong. Aminopectin does not have fructose. Aminopectin has alpha glucose. So aminopectin will be 1, 2, and 3, D. Trehalose is a sugar that gives a negative result when tested with Benedict's solution. A molecule of trehalose forms 2 alpha-glucose molecules when it's hydrolyzed. Interesting. So which row is correct? A formula of trehalose and then a sugar that gives the same result. Okay, so you have to understand here it's a compound of alpha-glucose. And when alpha-glucose is going to be condensed together, this means that it will lose 1 water molecule. So the formula is not going to be, C12H24O12. It's not like, it's like a carbohydrate that is specifically a monosaccharide where it has a formula of C2H2NO2. You have CnH2nOn. Okay, it's not this way around because again it's a compound, and so it would have lost a molecule of water, and a molecule of water will be H2O. So you have to subtract 2 hydrogens and 1 oxygen. So instead of being 24, it would be 22. Instead of 12, it would be 11. Now, because it gives a negative result, that reminds us of sucrose. And obviously it's the only other compound that we know of that is going to give you a negative result because it's a non-reducing sugar. So it's B. Okay. 10. Olestra is an artificial lipid. It's made by attaching fatty acids by condensation to a sucrose molecule. A simplified diagram of olestra is shown. R represents the positions where fatty acids would be attached. Okay, so R is basically where fatty acids would be attached. Interesting. And then he says humans cannot hydrolyze olestra. However, other animals may be able to do so. How many molecules of water would be needed to hydrolyze 1 molecule of olestra into fatty acids fructose and glucose? Okay. In order to hydrolyze into fatty acids, fructose and glucose, that's interesting. Again, it's basically made by attaching fatty acids by condensation to a sucrose molecule. And okay, now this is fructose. This is glucose, and R is supposed to be where any fatty acid chain would be attached. Wow, okay, so R represents the position where fatty acids will be attached. So we have lots of fatty acids, as you can see here. 1, 2, 3, 4, 5, 6, 7, 8. 8. So we need 8 water molecules to hydrolyze that, and then we need another 1 water molecule to hydrolyze this one. So that would be overall 9 water molecules to hydrolyze these bonds. Okay. Which molecule contains at least one peptide bond? Okay, one peptide bond. Now, obviously, we don't have any peptide bonds here in A because, again, what's a peptide bond supposed to be? It's supposed to be NH and then the connection between the C double bond O. Okay, so this is supposed to be what we're looking for over here. Now, we don't really see this in A. Do we see it in B? Actually, we do. We have this bond over here. We also have this bond over here. So we have 2, so that contains at least one peptide bond. You can see it's not available in C, neither in D, so I would say it's B. Okay, RNA polymerase and peptidyl transferase are both enzymes involved in protein synthesis. This one being involved in translation, this one being involved in transcription. Which statements describe similarities between these two enzymes? They're both globular proteins. That is true, of course, all enzymes are globular proteins, and they both have the same tertiary structure. That's wrong. Different enzymes have different tertiary structures. Remember, tertiary structures determine the shape of their active site. They can't have the same active site because they have different substrates. So this would be wrong. They both intracellular enzyme. That is true because they stay inside the cell. They function inside the cell. So I would say it's only 1 and 3. It's B. What's a feature of competitive enzyme inhibition? The inhibitor binds permanently to the active site. Okay, competitive enzyme inhibition, the inhibitor will bind to the active site. However, it usually does this reversibly, not permanently. That would be incompetitive inhibitors that would bind permanently to the allosteric site. So I would say A is wrong. Inhibition can be reversed by increasing the concentration of substrate. Actually, it's true. When you increase the concentration of substrate, the effect of the inhibition does decrease. The inhibitor molecule changes the secondary structure of the enzyme. It doesn't. The substrate and the inhibitor are the same shape. Okay, so no, they're not the same shape. They have similar shape, and that's why they're able to it's able to bind to the active site. Okay, so I'd say C and D are wrong, and B would be my answer. Batratoxin. Okay, sorry about that. Okay, is a poison found in frogs in the Colombian jungle. The poison is used to produce poison darts, and the poison works by increasing the permeability of cell surface membrane of nerve and muscle cells to sodium ions, which move out of the cells. 4 students made statements about how the poison affects the cells. Now, when sodium leaves, that means that the concentration inside the cells would decrease, meaning the water potential of the cell would increase. So water would leave the cell by osmosis, causing the cell to shrink. That is true. Water enters the cell. Actually, no. When the sodium ions move out of the cells, the intracellular fluid has a more positive water potential. That is exactly what happens when it's more positive water potential. That means it's a higher water potential. The reason for this is because when sodium leaves, the concentration inside decreases, and when concentration decreases, that means we have more water content. Okay, when sodium moves out of the cells, the extracellular fluid has more positive. That's the opposite, so I would say it's 1 and 3. A. Which processes use energy in the form of ATP? Endocytosis, exocytosis, facilitate diffusion. Obviously, cytosis, but not diffusion. Diffusion is a passive process, so facilitate diffusion cancels, and so it's 1 and 2 only. An indicator is colorless and acid and pink and alkali. An experimental petri dish of agar was prepared using an acidic solution of this indicator, and a disc of agar 1 centimeter in diameter was removed from the center to create a well. Interesting. So again, we have an experimental petri dish was prepared using acidic solution of this indicator. So basically, we have a petri dish with agar and basically some indicator inside, and it's an acidic solution of the indicator. Now, a disc of agar 1 centimeter was removed from the center to create a well. So now we have a well created inside here. So this is empty. Now, what's it called? A white card showing circular marker lines 1 centimeter apart were placed underneath a petri dish. Interesting. So we have a white card placed under the petri dish with lines on it, right? Okay. So he said here, 1 centimeter cubed of alkali solution was put into the well. So we have here alkali put into the well, and a circular disc of pink color appeared and spread through the agar, and it reached the first marker line in a short time, but took longer to reach the second marker line and a very long time to reach the third marker line. So which explains these observations? Facilitated diffusion of alkali, obviously not facilitated. There are no channel proteins in agar dish, so we could cancel out facilitated diffusion. However, it's obviously the simple diffusion of the alkali, because the alkali was the one placed inside the well, and it's going to travel from the well through the agar. And so here I would pick simple diffusion of alkali, of the alkali solution. 17. The diagram shows a section of glycoprotein molecule found embedded in a cell surface membrane. Each of the amino acids is represented by a small shaded circle. Okay, so we have the amino acid chain forms an alpha helix, and these are different amino acids, which are like small circles. So which row shows the property of the amino acids found in the alpha helix and the property of the amino acid Q? Okay, now the amino acids found in the alpha helix specifically are found inside the hydrophobic region of the phospholipid bilayer, because it's just basically filled with fatty acid chains. So yeah, I would say these are definitely non-polar. However, the amino acids that are on the outside would be surrounded with water and cytoplasm and everything. So yeah, sure, they are polar. Okay, so A. Some cells are listed: bacterial cells, cancer cells, lymphocytes, mature blood cells, stem cells. What cells can divide by mitosis? Of course, stem cells can divide by mitosis. Red blood cells can never divide by mitosis because mature blood cells do not have a nucleus, and the nuclear division is what we call mitosis. Lymphocytes can totally divide by mitosis. They can form plasma cells, which divide more and more. And then we have cancer cells, which obviously can divide by mitosis, and that's the reason they're cancerous, is because they divide indefinitely. And then bacterial cells actually divide by binary fission, not by mitosis. So I would cancel out 1 and 4, so it's 2, 3, and 5. That's C. Which statements about mitosis are correct? At the end of telophase, 2 nuclei are formed. So at the end of telophase, yes, sure, obviously 2 nuclei are formed. Centrioles attach chromosomes to the spindle during metaphase. That is true. The spindle fibers are attached. No, not centrioles. Centromeres are what the spindles are attached to. That's a point of confusion. I just got confused myself. Centrioles are organelles that form part of centrosomes, which are basically supposed to be these organelles that synthesize the microtubules and act as the microtubule organizing center, which basically are going to act as the spindle fibers. I hope this makes sense. So no, they're not the ones that attach to the spindle, to the chromosomes. That's wrong. Okay. Chromatids are pulled apart during anaphase. That is true. Okay, so 1 and 3 only is C. Which statements about telomeres is correct? They allow cells in culture from any age of donor to divide a fixed number of times. Okay. Telomeres are basically the end of the chromosome. So let's draw out a chromosome. So the chromosome would have these ends that basically contain non-coding sequences, specifically of G and C, guanine and cytosine. They're incredibly found in telomeres, and these non-coding sequences this is basically just to help the DNA last longer, because when when DNA replicates, DNA polymerase enzyme does not run until the end of the DNA molecule, and so part of the DNA is to be lost. So part is a mechanism to avoid this. We have telomeres, which are going to when the DNA polymerase does not run until the end, it runs until a part of telomeres. So obviously we wouldn't lose any part of DNA that we require that is coding. Okay, so they allow cells to divide. Obviously they determine the age of or the lifespan of cells. They don't really allow you to divide a fixed number of times. There are genes which are present on the 5' end of every chromosome. No, they're unpaired regions of DNA. That's not what they are. They prevent introns and exons from being lost from genes during cell division. I would say so. Introns and exons are supposed to be the coding and non-coding parts of DNA, and so I would say yes, it prevents the loss of any DNA during cell division, because part of cell division would be obviously the S phase. The S phase being the synthesis phase, this phase where DNA replication would happen. So I would say it's D. Okay, the diagram shows stages of mitosis. So we have 1, 2, 3, and 4, and 5. Okay, 1. we see what's it called? I would say obviously 2 is really obvious, it's anaphase, so that would be one of the last phases. 5 would be like early stage anaphase, middle stage like metaphase. So like it's obviously like you could see how they're like closer together. So that's also like part of anaphase. And then we have like 4, which is like, I suppose this is metaphase because they're all aligned together. And then 1, I would say, is when they started, I think 3 is when the nucleus just broke down, so this would be like maybe prophase, G phase, G2 phase, and then 1 it would be like mid prophase going on to like metaphase. So which sequence is correct? Wait, you can see here in 1, I mean they're obviously clear, but like okay, let's see. So I would say, yeah, it's 1 going on 3, going on what's it called? Well, 4 going on 5, going on 2, or going 2 going on 5. Let's see the options. We have 1 3 4 5 2, or 1 3 4 4 2 5. And we also have 3 1 4 5 2. Okay, let's see, let's figure this out together. Okay, now 1, we could see how like the chromosomes are like obviously it's just like very very thick. So 3 has to happen before 1 because 3 is the beginning of prophase, where like the nucleus just broke down. So I would say it's 3, then it's 1. Now 4 5 2, or is it 4 2 5? Let's see. 5 is anaphase, but 2 is supposed to be the end of anaphase. So it's obviously going to be 5 then 2. It cannot be 2 then 5. So I would say it's 3 then 1, then 4 then 5 then 2. So let's switch this. Which one is this? That's C. Okay, moving on. Which statements about transcription and translation of a gene is correct? The non-transcribed strand of DNA has base sequence that's identical to mRNA. Obviously not identical because mRNA, remember, is RNA, does not have the thymine base. It actually has uracil. So instead of T we have U. And the template strand, again, not identical, never identical. The non-transcribed sequence strand has a DNA, has a base sequence that's complementary to the tRNA molecules required in translation. That is actually true because let's draw DNA over here. So we have the non-coding strand, and then we have the template. We have the coding strand and have the template strand. mRNA is going to come to, like, let's say this is the non-coding / template, okay? mRNA is going to come here and it's going to form something that's complementary to her. Now this guy, the coding strand, is actually complementary to the non-coding or the template strand. And so since both mRNA and the coding strand are identical now, because obviously this guy is complementary. They're not identical, but you get the point. Like, they're both complementary to her. So now when when tRNA comes, when it's time for tRNA, okay, tRNA will be complementary. So tRNA will be complementary to mRNA and thus complementary to this guy as well. So it will be C. Okay, I hope this was clear. I'm sorry if it was confusing, the drawing was confusing. Which statement about mRNA is correct? The primary transcript becomes modified by joining introns to become mRNA. Actually, no, by splicing off introns we become mRNA. The primary transcript is synthesized and then modified to mRNA in the nucleus. True. mRNA contains nucleotides containing the sugar deoxyribose, of course not. It's RNA. R stands for the ribonucleic acid, where obviously we're talking about ribose with ribo. Okay, the bases in mRNA are held together by covalent bonds. The bases are held together. Their bases are not held together. The nucleotides are held together by covalent bonds. So it will be B. Okay. The diagram shows a part of a DNA molecule, and here it says which label is correct. Okay, so we could see here that we have 2 bonds, and then here we have 3 bonds. So obviously these are supposed to be C and G, and this is supposed to be A and T. Now, how do we decide which one is A, which one is T? Obviously A is the purine, which is supposed to be double ring structure, so you could see that this is a double ring and this is a single ring, so obviously this is thymine. Now here we have double ring and 3 bonds, so obviously this is guanine, and then single ring and 3 bonds, that's cytosine. Cytosine bonds with guanine, adenine with thymine. Adenine is double ring, guanine is double ring, thymine and cytosine are single ring, or in other words, pyrimidines. Which label is correct? Let's see. A is adenine, that's wrong, it's guanine. B is guanine, that's wrong, it's cytosine. C is cytosine, that's wrong, it's adenine. And lastly, D is thymine, that's true. The sequence of the bases in DNA coding for the first 8 amino acids in beta polypeptide of adult hemoglobin is given to you, and then however in hemoglobin C, which is the basic cause of hemolytic anemia, it becomes the following. And there's a change of bases you could see here from CTC to TTC. Some of the DNA triplets that code for amino acids are listed in the table. You have amino acids and you have DNA triplets, so you could see here how DNA triplet that codes for glutamic acid is changed into TTC, which actually codes for lysine. So we have a change in amino acids, and what change occurs to the amino acid sequence of normal hemoglobin to make it hemoglobin C? Glutamic acid is changed to lysine, exactly. The diagram shows a plant organ which letter correctly labels the xylem. Okay, we have obviously the xylem is going to be on the inside of the vascular bundle. Which one is the vascular bundle? This over here is the vascular bundle. Now this is the xylem. What are the others? C is the cambium, and B is the phloem. It's on the outside, and then A is supposed to be the pith. So this is why we choose D. The photomicrograph shows a vascular bundle found in a plant organ. Which statements about this vascular bundle are correct? Okay, so you have the vascular bundle in a plant organ, which I would guess this is a stem. So again, we've labeled this in the question before, but this over here is the xylem, this is the cambium, and this is the phloem. Now, which statements about the vascular bundle are correct? The vascular bundle is part of a structure of a root. I would not say so. It's actually part of the stem. Some of the what's it called cells in region X have very large numbers of mitochondria. Region X, I would guess the region X is between the cambium and the phloem, and some of these cells will be the companion cells of the phloem, because some of the cells will be part of the phloem. So I would say maybe yes, that's right, that's not wrong. Region Y is made up of a number of different cell types. Region Y, there are different cell types here. You could see there are smaller, less developed xylem forms, and then there's bigger xylem forms. So I would say yes, 2 and 3, which is C. Which changes to water potential and volume of solution in the phloem sieve tube element occur when sucrose is moving from the sieve tube element to actively dividing shoot tip? Okay, so water potential in the phloem sieve tube element versus the volume of solution in phloem sieve tube element. Okay, we're moving to the actively dividing shoot tip, and we want to provide it with minerals and amino acids and everything. So to do this, obviously water potential is going to increase because we're losing the material. So I would say it's higher, and the volume of the sieve tube element is going to decrease because again we are losing substance. So I would say higher, and it decreases. Which process occurs during the loading of sucrose into the phloem sieve tube elements? Protons are pumped out of the cytoplasm of companion cells into its cell wall. That is true. That does happen during loading. Okay, there's a higher concentration of protons in the symplastic pathway outside the companion cell. A higher concentration of protons in the symplastic pathway? Outside the companion cell. It's not really in the symplastic pathway, rather the apoplastic pathway, because it's in the cell wall. So I would not say so. However, let's see 3. Protons are unable to move back into companion cell. Again, that's wrong. Protons do move back by facilitated diffusion through cotransporter proteins. A cotransporter molecule acts as a carrier protein for protons and sucrose, exactly. So I would say it's 1 and 4 only. That's B. Okay, the photomicrograph shows a section through the structure found in mammals viewed using a light microscope. So we have here W, which is the second, and if not the innermost structure. What are the main components of layer W? Collagen fibers. Actually, collagen fibers are mainly in the final layer. However, the middle layer is going to mainly contain elastic fibers and smooth muscle, and based on the ratio of elastic fibers to smooth muscle, we would determine if it's an elastic artery or a muscular artery. So smooth muscle and elastic fibers, exactly. Okay, which statement correctly links muscular or elastic arteries to their function? The aorta is an example of a muscular artery as it transports blood from the left ventricle to the heart. Actually, the blood vessels closest to the heart, like the aorta and the pulmonary artery, are actually elastic arteries because they need to be able to carry things like elastic recoil to make sure that they adjust the blood pressure nearest to the heart, because the pressure there is quite high. But then moving away from the heart, the pressure starts to decrease, and so we have the muscles which were able to constrict or to dilate the blood vessels so that more blood or or less blood, and what's called they're able to regulate pressure in that way. Okay, so the aorta is an example of a muscular artery. That's wrong. Arteries further away from the heart are muscular, exactly as they transport blood at high pressure. Actually, no, it's lower pressure. Elastic arteries expand when the heart contracts and then recoil as the heart relaxes to maintain pressure, exactly, and that's known as elastic recoil, a mechanism you studied in chapter 8, where basically, yes, the heart is going to contract, the blood's going to flow through the arteries, and once it does, the arteries are going to expand. They're going to expand, and then they're going to contract in order to expand first in order to decrease the pressure because it's incredibly high. Then they're going to relax later on, which is going to allow them to contract, and so again increase the pressure. So this helps regulate the blood pressure. So it's, yes, that's true. C. Okay. The graph shows the changes in pressure that occur in the left side of the heart during one cardiac cycle. What's the heart rate in beats per minute? Okay, so let's see the heart rate in beats per minute. So obviously you could see how this, the heartbeat here starts and ends like in 0.8 seconds, and there's one heartbeat is 0.8 seconds. So what is the heartbeat and beats per minute? So now in 60 seconds, in 60 seconds, how many heartbeats will we have? Okay, so you basically cross multiply like this. So you're going to divide 60 by 0.8, and that's going to be 75 being the heart rate. Which events occur during ventricular systole? Atrioventricular valves close, muscle and the ventricle walls relax, and semilunar valves open. Okay, yes, during ventricular systole, semilunar valves are going to open. The muscles and the ventricle walls, of course, are not going to relax. They're going to contract, and that's why the semilunar valves will open, because they're pumping blood. They're contracting. And yes, sure, atrioventricular valves will close to avoid the backwards flow of blood to the atria. So I would say it's 1 and 3 only, which is C. Okay, next we have which reactions take place in the capillary of the lungs. Carbonic acid is formed from carbon dioxide and water. Okay, let's remember the Bohr shift together. So what happens inside the red blood cell is that we have firstly so we have a red blood cell, and it's supposed to be now in the capillary right next to the alveolus. So this is where the alveoli, and this is obviously the red blood cell. Now in the alveoli here, we're going to have lots of oxygen. Oxygen here is going to move down its concentration going to the red blood cell, where here it's going to react with hemoglobinic acid. So hemoglobinic acid, HHB, is going to react with oxygen, and this is going to get us back to hemoglobin, oxyhemoglobin, right? So we're going to form oxyhemoglobin all over again. Now the hydrogen ions that are going to be released here are going to react with the hydrogen carbonate ions from the blood plasma. Remember, they're going to diffuse back, and then that's going to form H2CO3. That's carbonic acid, and that is in presence of carbonic anhydrase enzyme. And then what happens later on is that obviously there will be the chloride shift. So since there are hydrogen carbonate ions moving in, there will be chloride ions moving out, and then lastly the H2CO3 is going to break down into CO2 and H2O, and then CO2 over here is going to move out of the red blood cell and into the alveoli so that it could be expelled out from the lungs. So carbonic acid is formed, that is true, but not from carbon dioxide and water, however from hydrogen ions and HCO3 minus. This is wrong. Okay, carbamine hemoglobin is formed from carbon dioxide and hemoglobin. Actually no, if there is carbamine hemoglobin, which obviously there is, okay, carbamine hemoglobin is we're going to have oxygen available, so obviously hemoglobin has a high affinity to oxygen, so oxygen is going to react with hemoglobin, and that's going to expel carbon dioxide, so this is also wrong. And then hemoglobinic acid is formed from hemoglobin and hydrogen ions. Actually no, hemoglobinic acid is not going to form, it's going to break, it's going to react with oxygen, and it's going to form oxyhemoglobin. Carbon dioxide and water are formed from hydrogen carbonate ions and hydrogen ions. Exactly. Carbon dioxide and water, this is what happens. So 4 is the only correct option here, so it's D. Which of the features are important for the process of diffusion of oxygen out of an alveolus? Blood pressure forces red blood cells through capillaries. That is actually true, because when there's pressure that's going to push the red blood cells closer to the surface, basically making them completely surrounded by capillaries, and so oxygen is free to diffuse with greater surface area to volume ratio and everything. 2, epithelium is permeable to respiratory gases. I don't mean surface area to volume ratio, just greater surface area in general. Now for 2, epithelium is permeable to respiratory gases. That's actually great. The epithelium is permeable, so I would guess Obviously, that's important for the process of diffusion. Moist squamous epithelium is present. That does actually allow the gases to, diffuse and turn into what's called aqueous materials, and then go back to what's called how they were. So, for example, this is also important. However, substance to reduce surface tension not only is to avoid alveoli collapsing, doesn't really have to do with oxygen diffusion. So I would say 1, 2, and 3 as A. Okay. A student viewed 3 slides at low magnification and high magnification. Each slide was a section through a different airway of gas exchange system. The students observed 3 features inside each slide. So the 3 features observed by the students are an irregular arrangement of cartilage, highly folded inner layer, cilia and epithelial cells, very few goblet cells, cilia and epithelial cells, and a thick layer of smooth muscle relative to the wall thickness, smooth muscle tissue, and blood vessels in many, many goblet cells. Okay, so let's see and let's identify how to label each one of those. Remember, guys, moving from trachea to bronchi, which basically, the bronchi, then moving to the bronchioles. And then lastly, forgot about the bronchus. And then we have, bronchus is really just a singular word for, sorry about that, I was just a bit dizzy. Okay, so it's bronchi or bronchus, singular plural, doesn't matter. And then alveoli. Okay, trachea here is going to have the greatest amount of cartilage, and specifically going to be C-shaped. It's going to have smooth muscle, it's going to have epithelial cells, it's going to have goblet cells, it's going to have ciliated cells, everything. Okay, bronchi are going to have exactly the same thing, except for cartilage, they're going to be fewer on cartilage and they're going to be block-shaped. Bronchioles, we're going to move here. Actually, we're going to start decreasing. We're not going to have goblet cells, or like a little like at the beginning of the bronchioles, we're going to have some goblet cells, then they're going to start decreasing until we reach the alveoli. So here we have no goblet cells, no ciliated cells, obviously, no nothing. It's just basically epithelial cells. But here we do have cilia, okay, and we do not have smooth muscle here, okay. So we stop at like smooth muscle like at the beginning, but then again it gradually stops. No cartilage here at all. So let's see. Okay, so we have an irregular arrangement of cartilage, highly folded inner layer, and cilia and epithelial cells. So I would say that would be the bronchi or the bronchus. Okay, and then very few goblet cells, cilia and epithelial cells, thick layer of smooth muscle relative to the thickness. So I would say this may be the beginning of the bronchiole, because like obviously we don't have like smooth muscle in the end of the bronchiole. We also have cilia, and we also have very few goblet cells at the beginning. So I would say it's bronchus, bronchiole, and then lastly smooth muscles, many goblet cells, blood vessels. I would say obviously it's a trachea, so that's A. Okay, which terms can be used to describe the role of mosquitoes in transmission of malaria? So it's not really they're not really the parasite. They're the vector. Okay, they're the ones that carry the parasite from one person to another. So they're not the pathogen. They're not the disease. They are the things that will carry it from one place to another. So it's D. Rheumatoid arthritis is a disease which causes the body's immune system to attack its own cells, and the disease can be treated using monoclonal antibodies. Now the table shows 5 different monoclonal antibodies and their modes of action. Then we have inflammation and swelling of joints are symptoms of rheumatoid arthritis. The cytokine TNF-alpha will activate the cell's immune system, leading to the death of cells in the joint. So here the mode of action of the disease basically is that we're going to have these cytokines activating the immune system, triggering it, and therefore this is going to lead to maybe killing of the cells of the joint. Now, which types of monoclonal antibody could be used to treat rheumatoid arthritis? Let's see. 1. Binding to proteins on cell surfaces and triggering the immune system. We want to avoid triggering the immune system, okay, because it is being triggered. It is being triggered, and so it's acting and killing these cells that are part of the body. So we want to stop this. Blocking molecules on cell surfaces that inhibit T lymphocytes. We are blocking the molecules that inhibit T lymphocytes. We want T lymphocytes to be inhibited, so we shouldn't be blocking the molecules that inhibit them. So we should cross this. Blocking cell signaling receptors that trigger cell division. It's not really a cancer kind of situation where we're going to stop cell division, because it's not really happening. What's happening here is that we have cytokines triggering the cytotoxic T cells, which are going to kill the pathogen, or kill not really pathogens, just the cells of the joint. So I wouldn't really choose this for the mode of action. However, we'll see the options later. Binding to antigens on cell surfaces and releasing a drug. This could potentially work depending on the kind of drug. And then blocking cell signaling receptors that trigger the immune response. I would totally do this. This is exactly what we should be doing. Okay, which types of monoclonal antibody could be used to treat rheumatoid arthritis? I would cancel any options with 1 and 2 specifically, And this leads us to the last option, which is 4 and 5 only. A person's blood group is determined by antigens present on their blood cells. Antibodies in plasma of a person who receives the blood can make blood transfusions unsafe. The table shows the antigens and antibodies of people with different blood groups. Okay, so you have blood being AB and O. Okay, so which blood groups can give to a person with blood group A? Okay, so remember, for someone with blood group A, only A and O can give to them. Obviously AB could not give. Now, I will provide a chart in my description for the blood groups that can give to each other to make life easier, because if you memorize them, you won't have to refer to the chart, which could be confusing. Okay, question number 4. Some of the events during the primary immune response are listed. 1. Phagocytosis of a foreign microbe by a macrophage and antigen presentation. Some T lymphocytes will become T killer cells, which will kill infected body cells. Then the T helper cells will divide mitosis to produce T lymphocyte clones, and a T helper cell with a complementary receptor binds to the antigens being presented. What's the correct sequence of events during the primary immune response? Well, I would say that first we're going to have an inflammatory immune response. So we're going to have phagocytosis happen, where the macrophage is going to engulf the cell by endocytosis, and then it's going to form what's called the antigen presenting cell, which is obviously going to be when the T helper lymphocyte is going to bind to its complementary receptor on the antigen presenting cell, which basically means that it's going to now be able to activate the other cells, like cytotoxic T cells, maybe help them divide, maybe help form them, and the B lymphocytes, but that's a different immune response. So now we have 1 and then 4 is going to happen. Now between 3 and 2, what happens in 3 is that the helper T lymphocyte divides by mitosis to produce T lymphocyte clones, and then some T lymphocytes will become T killer cells and kill infected body cells. Yes, well, I would assume that here the divide mitosis produce these clones, and then some of these clones will signal or help develop the T killer cells. So it will be 3 and then 2, so that would be B, and that's it. So thank you guys for watching this video. I hope this video was helpful for you. If it was, make sure to like and subscribe so that this video could be shared to as many students as possible. If you have any questions, make sure to write them down and I'll answer them as soon as possible. I'll see you guys later. Goodbye.